So, yeah, I think we can start now that we've got ourselves coordinated. So we had to move around a little bit so that you could see the slides. Hadn't quite thought about how this panel discussion was going to work. But welcome and thank you for joining this session on integrated CLD, manufacturability to GMP-Ready by design. My name is Campbell Bunce. I'm the CSO at Abzena. And I'm joined by my colleague John and associate Scott, who will introduce themselves when they come up to the podium. But the format of this session is that we'll present some slides just to frame out the discussion and then move to the discussion and Q&A. By way of introduction, Abzena is a global CDMO and CRO partner with integrated operational sites across Cambridge in the UK, San Diego, and Bristol in the US. And we combine discovery, development, manufacturing, analytics, and regulatory expertise under one coordinated network. Our area of specialization is biologics and bioconjugates, particularly the next generation molecules that we're seeing a lot of excitement around, including bispecifics, multispecifics, antibody oligoconjugates. And we've got a number of technical platforms that we use to help our clients successfully develop their drugs. And one of those platforms is the topic of today, our cell line development platform. Now, at the core of success is this aspect of developability, which helps to de-risk drug development during candidate selection. And the goal is to essentially develop a kind of holistic understanding of the molecule. And we do that through answering two key questions. Can we make it and does it work? Can we make it considers things like stability, quality, scale, cost, time. And does it work considers things like mode of action and patient safety. And we have and to do that, we evaluate our candidates across kind of three key parameters. So we look at binding and function, immunogenicity and safety, and manufacturability. So this allows us to select, the candidate with the least risk to move forward. It anticipates manufacturing risk, and it reduces some of the longer term development issues that we might see and have to deal with at the more costly side of development. So manufacturability is the most important assessment when we are selecting drugs and for the future development success of that drug. And this really tests how the molecule performs in real development and manufacturing conditions such as thermal stress, low pH exposure, formulation stability, freeze thaw, and high conc viscosity conditions. And the data informs us of any stability, degradation, aggregation, formulation risks early, and allows us then to, as I say, select the best lead candidate with the de-risk profile to move forward, or indeed it might actually highlight that we might need to re-engineer the molecule. But what I haven't included in this slide is productivity. And of course, it's really important to understand if we can express the molecule that we want to manufacture on a production cell line platform. And when we look at the kind of typical journey of a drug, development journey of a drug, we tend to do things in sequence from discovery through proof of concept, through design developability, lead candidate selection, then cell line development into process development, scale up and manufacture. However, if we include cell line development during the developability process, this offers a lot of advantages, not least time, but also to ensure success that we can make the molecule in a production platform, and they can make it to a good level of productivity and quality that we need to take to GMP. So my colleague John is going to talk to you about our cell line development platform and the benefits that that offers when we link developability, cell line development and manufacturing planning with selecting better drug candidates, de-risking development, and actually getting drugs to patients faster. John?
Hello, everyone. So my name is John Gill. I'm the Vice President of Cell Line Development and part of the scientific leadership team at Abzena. And prior to, I think, going into the conversation that Campbell led with, I'll talk a little bit about the platform just so we know what the foundation is and where we're starting from and how we apply that to the developability or manufacturability conversation. Ideally, what we're doing is setting up these evaluations and these pools that are allowing us to, without risk and with confidence, move forward through the development and get off the critical path. I'll talk a little bit about that in a few moments. The AbSelect Pro workflow is from a higher level. The vector construction is initiated from your primary sequence using the proprietary 2G unique vector. It's from a small Dutch company, Proteonic, and it is based on GS selection. We work primarily with a CHO K1 GS system. With the use of the Proteonic vector, initiate transfection to generate your stable pools. This is really where we're going with a lot of the conversation, is reliability of your stable pool. There's some diversity, as in all stable pools. It's not just your clone, but that diversity tends to be a readout of tighter differentiation. The product quality that we're seeing at those early stages is very consistent, so it allows us to generate early material for all of your process development activities to, again, and if I say it another 12 times, I apologize, but getting the cell line development off the critical path. I've been doing this for a long, long time, and you get tired of being the leader of the cell line development function or just part of the cell line development function and having the eyes of the organization looking at you waiting for when's my turn. And especially when you're working with clients, how you can accelerate their development pathway. From that stable pool, we're generating 3 to 5 grams per liter. So with small amount or small volumes, whether it's shake flask or 2-liter bioreactors, that material can be shipped throughout the organization to initiate process development and other activities. From there, having that as part of the manufacturability platform, now you have the chance to almost even pause your cell line development, allow these other functions to establish proof of concept, establish your lead ID, and if you find that hit and you're confident in moving forward, going through the standard CLD platform, making sure that it's regulatory compliant and friendly, and go through your single cell cloning stages. For that platform, we're using the cytomine for the individual isolation of the single cell, and then we're using the cell metric from Salentum. Now I believe Nova for the image capture and submission of the monoclonality component of your filing. Within 10 weeks of the initiation of the transfection, we have your RCBs available, and that allows us to ship that again through the organization, whether it's within the Cambridge site for material generation to initiate analytical method development, or it's transitioning some of the material for formulation and getting that rolling so that you can, if it's an ADC, and we have the end-to-end capabilities, that you're doing your ADC process development with representative material. You can then rely on the single cell cloning just for your confirmation and lock steps. From those initial RCBs, we will run an AMBER 15 for the identification of your lead candidate. We can and do 2-liter or AMBER 250s for additional characterization of a lead and a backup clone, so that you have a little bit more, I think, resolute confidence in the candidate that would be used for your eventual scale-up for GMP manufacturing. And then ideally, and why we have confidence in the proteonic vector, is we initiate the stability study with our lead and backup candidates, up to 5 individual candidates for a 60 population generation stability study. And what we've seen thus far with this combination of our host and this vector system is greater than 95% of our clones have been stable. So with each individual client that we have worked with, there's a little bit of optimization that's required. This isn't just a monoclonal antibody industry any longer, so we do have anything from a cross-mab bispecific or some Frankenstein-like FC fusions that come through the shop. And what that requires, again, is confidence in the base host system, both the cell and the vector combination. So again, using the 2G unit vector, it confers higher rates of transcription. It's optimized dual promoters. But ideally, what we're doing in some of these stages for non-monoclonal antibodies is generating these vectors in a monocistronic manner. And what we're doing then is a lot of the chain ratio activities to make sure that you're getting the heterodimer that you're looking for, and that the product quality coming out of your initial stable pools is very high and very consistent. I think consistent is more likely what you're looking for when you're initiating the leveraging these stable pools to establish all your method development and process lock activities. So what we'll do there is generate multiple monocistronic vectors. We will do up to 3 stable pools for each of the combinations that we're looking at, and we'll do product quality in the embedded analytical team. So there's an analytical subfunction within each of the individual sites, which will facilitate method development and method transfer, so that's not something that we would lose additional time in terms of transferring your individual samples or your process pieces globally or to external partners. Here, and it's a critical part, is we will assess your, not just your titer, but we're looking at your growth profile and making sure that being under one shop, that we're very consistent in how we select the cell lines, because then we're really de-risking what clones are going through the manufacturing and GMP scale-up activities. Eventually then selecting the optimal pool, optimal cell line for scale-up activities. And really here, this is an image that, while you can look at, it's much more, I think, beneficial to have the conversation, because what we're trying to convey here is the reliability of the stable pool at these very early stages, and really in relying on the product quality consistency to allow that mat gen for all your process development, formulation, analytical method development activities, and then in parallel, generate your tox manufacturing material with the reliability that it will be consistent and there won't be excessive bridging or comparability studies for your eventual GMP material. So what we would do is the opportunity to work with the clients to generate your tox manufacturing with the stable pool while we're simultaneously doing the single cell cloning and cell line selection. What this does, from me personally, is getting cell line development off the critical path, but it's also accelerating the rest of your development so that when the GMP master cell bank is released, all of your processes are locked and confirmed with the initial stable pool activity, and it comes together for GMP manufacturability and reliability of your GMP scale-up. So really what we're trying to convey to all of you in initiating some of the conversations is that Abzena advantage, having the end-to-end capability where we're really identifying the initial risks. A lot of the time, the cell line development is considered that handoff from research to process development, or it's a very early process development activity, where having reliability of these early stage materials, it's an activity that you should add in your developability stages prior to your proof of concept, and having this cell bank with confidence in the freezer. When you've gone through your lead characterization and you've looked at maybe your top 5 molecules, it really de-risks that critical path for isolating the single cell cloning, getting to your MCB, but also linking all of the subsequent activities, those downstream activities going forward. So you're really de-risking your critical path with representative material. You're not relying on the transient to initiate some of the early screening. It's representative process and material, so there's not going to be significant changes other than confirmation and lock with your final clone to move forward. So what that does in reality, you're identifying those risks very early, and by streamlining the process, it's much faster to your drug substance. Eventually, and ideally while we're working within this process, we've developed our scale-down model to select the right clone for that initial scale quality. So whether you're going to 50 liter or 2,000 liter, the way that we're working from the get-go in your manufacturability, we have confidence that the clone selected won't require additional upstream process development, or it won't require additional 3 months of time to make sure that your upstream process is ready to lock and go to enable your IND filing. So with that, I think we can pass on to Scott.
Thanks, John. Hi, I'm Scott Rudge. I'm a consultant with Syner-G Biopharma Group based in Boston. I'm personally based in Boulder, Colorado. I'm also a member of Abzena's Scientific Advisory Board, and they were kind enough to ask me to participate in this and then to take the under on everything that they talked about. So earlier I talked about developability in a positive context. Now I'm going to talk about when developability fails. And the take-home message is going to be you really have to understand your biology. So where does developability get messed up? I'm going to talk about 3 case studies. One where there's a self-association that was favorable for the molecule. Another where dimerization was required for the mechanism of action. And then some interactions between freeze-thaw and viscosity where you might fall into a little trap. So I showed this slide again earlier. Some of you were here, not all of you. But there's a paper by Jane et al. in Proceedings in National Academy of Science in 2017 that described, I think it's 10 assays and grouped them into 4 groups that you could deploy on your newly lead candidate and predict whether it would be a provable molecule or not through manufacturing. And most of these assays focus on self-association and hydrophobicity or lipophilicity. And the thesis is that monomers, highly soluble monomers, water-soluble monomers get approved and other molecules don't. And they went through all these assays, and in those groups, if you have one assay that doesn't meet the threshold that they identified, that's a flag. And then they looked at which of those molecules were approved, and the vast majority of them, not the vast majority, 63% of them or so, didn't have any flags. But 15% of them had 3 flags. 3 out of those 4 groups, they failed to meet the threshold. So it's not a death sentence if you get a flag. They also showed from phase 2 and phase 3 how the molecules with more flags seemed to dropped out of. So it's a great study. It's really instructive, and if you haven't read this paper, it's a really good paper to read. But they cloned all these molecules into IgG1s, so they're not necessarily in the format that got approved. And this is all monoclonal antibodies. This is not bispecific. It's not fusion proteins. It's not anything else. So these guidelines are for monoclonal antibodies. I'm working on a project right now where the first thing we screened a number of candidates. We had one candidate that is represented by this top figure on size exclusion chromatography. You can see very clearly that there's 3 different species there. A high molecular weight, a higher molecular weight, and maybe the monomer is a little shorter on the side. There was another molecule that we screened, just 3 amino acids different from the one on the top. None of that behavior, single peak. And we told the sponsor, don't develop the top one, just stick with that bottom one. This top one's going to be a nightmare to purify or to work on. The sponsor ignored our advice, as he was right to do. And the top molecule gave the top 2 lines in this PK curve. So the graph on the right is the PK in a rabbit animal study, and the whole idea of developing this molecule was to have longer PK, is to be a longer acting drug, and you can see out by day 28 that the top 2 lines are 10 times higher than the middle ones and way higher than the comparator, which is the green one. So why is this happening? And our answer is we don't know. I mean, this behavior could be the property of the molecule that makes it have a long PK. It might not be. It might be unrelated. I I can tell you we expressed this in Heck, we expressed it in Cho, we expressed it at Wuxi, we expressed it at Wheeler, we went all over the place. Every single time we expressed this molecule, it gave these 3 peaks. And then we also learned if we did the size exclusion at pH 3, 1 peak. So it's some kind of ionic self-association. Don't know if it's important, but it gave us the right answer in the animal study, so we're not going to change it. And then we met with the FDA. We had a pre-IND meeting, and the FDA said, yeah, there's no rule that you have to have 1 peak in size exclusion. You just have to have something that's consistent and under control. And we think this is actually an equilibrium self-association, because if you take the same material that at pH 3 gives 1 peak and you titrate it back up to 7, you get 3 peaks again. So it's some kind of reversible self-association or something like that. And it's perfectly acceptable to the FDA. Would it be a flag in Jane's? Yeah, absolutely, this is a flag. So we have another molecule under development that its role is to keep two cell surface receptors from dimerizing. And it turns out in vivo, the target protein is a dimer when it prevents the dimerization of those cell surface receptors. The monomer doesn't work. So we made the dimer, and we made it as a fusion protein to the FC portion of an IG, and that was super active in the animal studies in rats. But it showed aggregation. It was something like 70% aggregate and 30% monomer. And when the CEO was trying to sell this to partners, the partners all said, we don't want to touch it. So we expressed it as a fusion protein to human serum albumin. We expressed it as a dimer to human serum albumin. This diagram's not exactly right. We did target protein to HSA. It wasn't a cyclized loop like this diagram is showing. But all of those super inactive in rats, like no activity whatsoever. So we're going back to the fusion protein, to the FC. We're going to deal with the aggregation, and actually we're going to try to make it more aggregate. So instead of 70% aggregate, we're going to try to get to 90% aggregate, because it's not really an aggregate. It's a dimer of this product, and that's how this product works. So again, red flag in Jane. Yep, wouldn't develop this molecule if you were following Jane, but the biology demands it. The biology demands that this molecule be present as a dimer and not as a monomer. And then finally, I just had this experience with a client that's following very strictly the Jane developability assays, and they did freeze thaw and they did viscosity, but they did the viscosity determination in an acetate sodium chloride buffer, and they got really nice viscosity, 150 mg per mil. They were around 12 centipoise. But they did the freeze thaw in a histidine sucrose buffer, and after 5 freeze thaws, they only saw the aggregate go from 1% to 1.5%. So they said no freeze thaw issues. But the problem is the buffers are designed, one to give the low viscosity and the other one to give good freeze thaw stability. So is the molecule itself compatible with freeze thaw, or is it a low viscosity? You have to pick the right buffer, right? And if you're picking buffers that are going to give you a favorable answer, then you're not really doing the test right? So when we scale up on this one, they're going to get into a pre-filled syringe, and then they're going to say we don't need freeze thaw. So the freeze thaw was really not as necessary. We're going to be in the acetate buffer so that we have the low viscosity, so we can do sub-Q administration. But again, would you get a red flag? Well, here Jane wouldn't give you any red flags, but they probably should, or they should say you should do the freeze thaw in the acetate buffer, just to make sure you don't have a freeze thaw issue if you do have occasion to freeze thaw this molecule. So those are my 3 case studies. I'm thanking you for your attention, and going to turn it back over to Campbell. I think that's it, right?
this is the part that's the panel discussion in the Q&A. I could probably sit down for this now because my head's not going to be in the way as much for you guys to see the slides. Thanks, Scott, for trying to undermine the the developability or the value of developability. But I actually think that it reinforced it, because for us, certainly at Abzena, as I mentioned earlier on, we look at developability in a very holistic way. We try and capture as many data points as we can across those three things, binding function, immunogenicity, safety, and manufacturability. And some of the examples you presented, I've seen things like that in the past, and it comes down to, I think, really understanding the mode of action, as you say, the biology. But then you need to connect the kind of structure to function. So, in the past, I've developed molecules that are, ligand or receptor fusion molecules, particularly in the TNF receptor superfamily molecules. These form trimers on the surface of cells. So when you make a fusion protein of them, sometimes you get trimers of dimers, at least that's what we saw. And to your point, as long as we could manufacture consistently and at scale, and we could characterize what we had, then that was fine.
Yeah, I think that. So thank you, Campbell. So I didn't undermine the whole bedrock of developability, but I think part of my point was I've seen people developability the potency right out of their molecule, and that's obviously what you want to avoid, right? It really should be potency, developability. Or actually, you need both. Really, you can't have just one. But if you don't have potency, you don't have a product.
Yeah, I agreed. And when you put those two questions about developability, can we make it and does it work? In some ways, it's in that order for a reason, because if we can't make it and it works well, then we've not got a product. But to your point, if it doesn't work but we can make it, same thing. So, but I think overall manufacturability is going to trump potency, because I think we can sacrifice some degree of potency for scale, consistency, cost, and everything else. But it needs to be looked at holistically.
Yeah. I agree. You can sacrifice some potency to manufacturability, but and maybe I'm belaboring the point, but I think the case studies I showed were cases where you just had to ignore that particular aspect of developability. Yeah. You had to say, I'm going to take the aggregation. I'm not going to try to get down to a single peak in my manufacturing. I'm going to let it go to the state that it wants to be in, rather than try to force it to be something that it's not.
Yeah, no, agreed. And I think a large part of this comes with experience as well. So over years, nowadays we're seeing some of the old drugs that we tried to develop and failed coming back. The multi-specifics, building in that kind of Swiss Army type function into one molecule. So that's when, paying attention to what you presented is really important. I agree. I don't know, have we got any questions that we can take?
Yeah, absolutely. So I believe Abzena received some audience questions earlier in the week. Can you hear me okay?
A little bit now, yeah, that's better.
Great. I believe at your booth you received some questions earlier in the week, so just to kick the discussion off, I'll start with those, if that's all right.
Sure.
So we had, how does the selection of your candidate clone change within an integrated platform?
So ideally, when you have an integrated platform, you understand what the media requirements are. You understand what the scale restrictions may be, and whether that's shear, pH, a number of different challenges within the operational process. So if you're selecting with that in mind, or you've developed more importantly, you've developed the host with that scale process in mind, you're de-risking the clonal selection. Typically, if you're bringing in a cell line that hasn't been optimized for what your eventual scale will be, that could trigger a number of upstream process development activities, which then will trigger additional characterization, which all in all will end up with time and cost. So ideally, with under one shop, it's been tailored from the start to fit to the end product, which is that scale and GMP manufacturing.
Does that change depending on what molecule you're working with?
Ideally, no, because you've done the developability up front. So you have the initial host and vector combination. So if you've identified within that stable pool that you can express the bispecific or your challenging molecule, it should be representative. And most times it is representative of what's going to be going on at scale.
Thank you. Another one you had was, so you don't use transposase currently, but is that something that you're thinking about using?
Great question. The short answer, yeah, absolutely. It's something that we're always looking at, whether it's transposase or other technologies. Ideally, from a cell line development platform, you have a couple of things in mind. You want a delivery of high copy number. You want to have the integration site be stable. And ideally, if you've identified those two things, if you have a transposase or not, you've established, I think, a quality platform. So while we are evaluating different transposase and internally developed as well, we're very happy with the titer, the product quality, and especially the stability that has been enabled with the proteonic-based vector.
Yeah, I mean, I'd just like to add that we have done extensive evaluation of transposon transposases, and for the moment we've not seen that it's improved over our, what we call our Abzena Pro. The proteonic vector plus our host line works extremely well in terms of the criteria that John's described, productivity, stability in particular, quality of the molecule as well. And we find that it seems to be very agnostic to the types of molecules. We've, vanilla antibodies, we get, extremely high titers, and with some really funky fusions, crazy fusions, we get really high titers, which is translated into manufacturing scale as well. So it's that kind of thing where, do we do we move and use transposase for perception or, the science? But to John's point, yes, we continue to evaluate transposase to see if it actually adds something extra onto our Abzena Pro platform.
Have you looked at stability?
Say again, sorry?
Have you looked at stability?
With the transposase?
Yeah.
Yeah, it's inferior to our Abzena Pro, at least the ranges of transposases that we've tested.
Fantastic. And then looking at the regulatory side here from someone else, how do you manage the regulatory challenges that next generation drugs can present when there's so little IND precedent?
I can start to answer that. We know that we're in a kind of new era, a new exciting era of drug development, where we see with antibody oligoconjugates in particular, where Avidity and Dyne have really blazed the trail for that type of approach. They're both at BLA, and we're excited to see the pipeline or fast followers of antibody oligoconjugates coming through. But the regulators don't have a lot of experience with these. They've not seen this type of molecule very much in the past. So what we find with our clients when they're developing molecules like that, kind of first-in-class molecules, is that we want to help the regulators understand the connection between structure, function, PK, and safety. And that's where the developability actually plays a large role, because you generate a lot of data during that process that helps us understand the molecule, what it looks like, what it should look like to be potent, to have the appropriate PK, to be safe and efficacious. And by providing the regulators with that data and try and fill in the gaps, and if we can't fill in the gaps, we try and look at parallel molecules or kind of similar examples to provide some degree of information. but taking the regulators with us in that journey rather than allowing them to ask questions and create doubt around what we know and understand about our molecules is not helpful. So taking the regulators on a journey with these new molecules is really important.
I'll double down on that, Campbell. I have yet to have a client in over 20 years who didn't think that their molecule was special and unique and first in class, and that the regulators just didn't understand them. So really is important to have a conversation and think about that you're educating them. You're helping them see your point of view. You have to give them a reason to say yes. So the burden is on you, and really any predicate that you can find to your molecule, even if your molecule is truly first in class, there must be something like it. There must be something that you can compare to and say, it's not completely different. Let me help you understand it if you understand it in this way, with this comparator or something like that. It's very helpful when you're having your discussions with any regulator, really, not just U.S. or the EU, but any regulatory body.
Brilliant. And now, well, I'll open it up to the live audience. Does anyone have? Yeah.
Okay, Campbell. I want to know about your progress with EpiScreen assay. EpiScreen, immunogenicity prediction. What's the news?
EpiScreen is part of the developability platform technologies that we use. Abzena was kind of innovators in this kind of clinical immunogenicity risk assessment. So we've got a suite of assays that we use. So we've got bioinformatics platform to look at the sequence, to look for any kind of immunogenicity liabilities there. And we continue to improve that in terms of reliability and speed and predictability. But we've also got the ex vivo assays where we use primary human peripheral blood mononuclear cells. So we've got an array of assays there, but we've advanced that assay so that we can actually, generate a lot of data in the one pot. So we can look at, all different types of immune response, in the T cells, CD8, CD4 T cells, and the profile of those responses and the strength of those responses, so that we can actually start to fully characterize and better predict immunogenicity risk, so the likelihood of these new molecules inducing anti-drug antibodies in the clinic. So the richer the data, the more kind of points you've got to actually build a correlation between what we see ex vivo in the lab and what we're likely to see in the clinic.
Lovely. Does anyone else have any other comments or questions on what's been discussed so far? Lovely. Did you gentlemen have anything else you'd like to add?
No, just to say that we know that more and more we get the pressure to take new drugs to patients as quickly as possible. So, at Abzena, we're lucky to, be exposed to and help develop these next generation molecules. And we do endeavor to develop them faster than, antibodies. But there is a reality around that as well. But again, I want to go back to that developability side of things, because the more we do there, the more we understand our molecule, the more data we generate, and the more we kind of make efforts to de-risk at that early, more cost-effective stage of development. That pays dividends downstream. So, paying a bit more attention to that front end, prevents, very expensive fixes when we get to the expensive manufacturing and clinical study stage.
I have a question regarding using AI to predict the behavior or the selection of clones or whatever the softwares that they say, okay, these antibodies are working very well, but when you translate them to the lab, they don't work very well. So do you have any input on that front?
So I know that there's at least one, I'll say pre-development company out there that's offering an AI service that's based on the Jane panel of assays. And they've taken that even further and into thousands of sequences, and tried to make predictions based on the sequence with AI and how they're going to perform in those developability assays. There's at least one that's done that. It's fascinating, I think, but I think there's also a question of whether it's real. AI is great. I like to say it's great at interpolating and not so great at extrapolating. So if you're looking at two things in between cases that are already known, AI is really good at telling you what happens in between. But if you're trying to extend from those cases, AI is pretty unpredictable. Or I shouldn't say it that way. AI does what it does, right? AI tells you the answer that it's learned or been trained to predict. Just the quality of that prediction when it's an extrapolation is not as high as the quality of the prediction when it's an interpolation. And so understanding whether you're extrapolating or interpolating and understanding that the AI is telling you with great confidence because it's always going to give you like very discrete, confident answers. It won't. You have to decide for yourself how much confidence you should have in that confident answer.
Yeah, I would say, yeah, I think it's early days for AI. I mean, if you look at that famous example where they used AI to look at histology slides to predict which histology slide had cancerous cells in it. And AI told you it was the one with the kind of measuring stick next to it to measure the diameter of the tumor. So, you know, it depends on the information it's getting, you know, through machine learning into AI. I mean, with AI, I think what we can do, we can screen things a lot faster, or we can look at sequences and get to an optimal sequence faster as well. So we've got within our developability, design to developability sort of platforms, when we're looking at optimal sequences in antibodies, so immunogenicity, aggregation liabilities, you know, deamidation sites, etc., and optimizing that sequence, we don't necessarily use AI, but we use it to get to the optimal designs, or at least down select more quickly. So we used to spend 2 or 3 days doing this, but now we can do it in seconds. So it helps do things quicker. So if we can do that more quickly and put more sequences through to down select and then take to wet work, then that saves a lot of time, money, and effort. So I think we'll get there with AI eventually in terms of being able to design drugs. I mean, I should say also there's companies out there that use AI to design antibodies, from de novo, or improve affinity. At the moment, I mean, we've worked in partnership with some of these and we've not got it to work as well as the old-fashioned way. Not yet.
Have any more audience questions? Scott, do you have a final comment for the audience, key takeaway?
One, I'd like to say these mics are very unidirectional. So you have to point them right at your mouth in order to they should make them solid on the side so you don't think that you... but second, what I'd like to say, and this is going to force Campbell to say something else, which is my goal. I've always thought of Abzena as a manufacturing company, a CMO, but really Campbell's organization over in the UK has so many capabilities in immunogenicity screening and all kinds of assays that they can do. And I think that's a side of Abzena that not many people are familiar with. So I want to invite him to talk a little bit more about the capabilities in Cambridge.
No, thanks for that, Scott. That wasn't a plug. That was that was off his own back. But so Abzena, I mean, we've been building Abzena for the past, I would say, 25 or so years, 25 years. And I joined Abzena in 2015. And we were a UK-based company, and we were a kind of early stage design developability or design development company. But when I joined, I came with 18 years of sort of biotech experience. And it was clear that what we had within that front end of our business was a kind of assembly of expertise that really addressed a lot of what biotech were needing, especially, startup emerging biotech, what they were needing to actually help and facilitate, get them through those early value inflection points and, generate data to go to investors to raise the capital. To then move their drug forward, and most importantly, move their drug forward through manufacturing and into patients. So that's how we've built this business, from the front and then through to the back in manufacturing. So we acquired businesses through 2015, 2016 in the US to then build that manufacturing footprint. So, I mean, we are known for being able to manufacture, biologics, bioconjugates, linker payloads. But I'm pleased that Scott recognizes the importance of that front end of the business, which really helps clients, reduce the risk of them being a victim of the valley of death, and then getting through those successful, painful points through manufacturing into the clinic, as I say.
John, will you close us out?
Dangerous proposition. I think I'd just go back to the intent of the conversation, and what I would do from my experience at pharma is really just, if there's no change in the cost or the effort, that to really rely on the stable pool as early in your developability conversation as possible. Stop with the transient and hope approach and really apply a reliable system very early on in the developability. So when we're looking at the Jane studies, and beyond, those materials need to be predictive. They need to be consistent, and they need to be something that, and if I go to my formulation team, they're not going to have a lot of confidence, and they're not going to probably rely as heavily on the data outside of just a regular thumbs up or thumbs down, unless they believe in the quality and consistency of the material. So it's really prevent the old handoff, throw it over the fence, research to process development or CMC, and really make it a much more integrated and thoughtful process very early on. And that's it.
Can we please thank our panelists?